Huntington's disease is a devastating genetic disorder that typically affects individuals in their 30s and older. Huntington's symptoms include spastic movements, loss of physical control, a change in mental state, and the eventual inability to communicate or control movement. These symptoms occur primarily due to the death of neurons in the basal ganglia of the brain. Basal ganglia are thought to control motor function and be involved in sensory processing. The death of these neurons are initially triggered by an abnormal Huntington protein in individuals with the disease. The Huntington protein is produced by a gene found on everyone's fourth chromosome. The non-mutated gene ends with a sequence of DNA bases, cytosine, adenine, and guanine, or CAG, which codes for a particular amino acid on the protein. However, in the mutated gene, the CAG sequence repeats itself multiple times, resulting in a mutant protein with an elongated tail of that particular amino. Unlike its wild-type or normal protein counterpart, the mutant Huntington protein interacts with the neuron in many dangerous ways. The mutated Huntington protein is responsible for destroying neurons through a number of different mechanisms, of which we will highlight 3. The addition of channels, the formation of aggregates, and damage to mitochondria. Mutant Huntington interacts with certain channels called NMDA channels in the neural membranes. Firstly, mutant Huntington pulls more NMDA channels than normal into the neural membrane. The higher number of channels causes a higher influx of both sodium and calcium ions into the cell. The increased number of ions overstimulates the neuron and damages it. Mutant Huntington also binds to a number of proteins also present in the neural membrane next to the NMDA receptors and activates them. After activation, these proteins can run around the cell and activate other factors that cause cell death or apoptosis. The higher amount of calcium allowed into the neurons by the high number of NMDA receptors also leads to the activation of enzymes called caspases. Caspases are in charge of chopping up proteins in the neuron. In Huntington's, the higher levels of calcium activate more caspases, which run around the cell and cut apart the extra long mutant Huntington proteins. The mutant Huntington pieces then clump into clusters of protein fragments called aggregates. These clumps then trap other important factors in the cell. These aggregates can travel into the nucleus of the cell and trap proteins that regulate gene expression. For example, when a particular protein that works with a gene for BDNF is trapped by the aggregates, it is not able to properly regulate the production of BDNF, a necessary protein. As a result, the cells have lower levels of BDNF than a healthy cell, which can then lead to cell death. Another way mutant Huntington harms the cells is by interfering with the proper function of their mitochondria. Mitochondria make energy in the form of ATP for the neuron, and when they are not working properly, the neurons cannot produce the energy needed to survive. Normally, mitochondria split apart through fission and then join together again through fusion. This process is regulated by many proteins, including the DRP1 protein, which regulates mitochondrial fission. Mutant Huntington binds to DRP1 and leaves the mitochondria unable to regulate fission, so they divide more than they would normally. This mutant Huntington and DRP1 interaction also makes it more difficult for the mitochondria to fuse back together. The imbalance between fission and fusion causes the mitochondria to break apart and lose proper function, which means less ATP and therefore less energy for the cell. In addition to these highlighted issues, the mutant Huntington protein can cause many other problems associated with the disease. Scientists will continue to investigate the many ways that mutant Huntington contributes to the pathology of the disorder. While there is no cure for Huntington's, a number of therapies are being investigated that may help patients with certain symptoms of the disease, including the motor difficulties and mood changes. Occupational and psychological therapies have proved effective in improving day-to-day life for Huntington's patients.